afternoon. My name is Victor Antontsev, and as mentioned, I'm the head of Biosystems Engineering at VeriSIM Life. So today, what I'll be talking about is BIOiSIM, and that's the software platform that we've built internally to accelerate drug development across preclinical and clinical stages of compound testing using our internal and proprietary computational hybrid modeling approach. Now, to get into the specifics, and this is a problem that I'm sure that everyone here has experienced at some stage in their development. In the current paradigm, preclinical research is expensive, slow, and doesn't act as a good filter. Success is pretty low once compounds actually make it to clinical trials. So just over 90% of compounds at those phases will fail for toxicity or efficacy reasons. And in silico approaches, computational approaches, have been applied to the drug development pipeline overall, but they tend to be focused on the drug discovery side, finding new targets where they leverage tools and novel technologies like machine learning and deep learning. Or in the later stages of drug development, these models tend to be very overfit. So there will be one compound and one model customized to that specific compound, or these models will be oversimplified. They'll look at the body as a system of one or two different compartments, and this type of insight isn't actually translatable to a lot of really specific insights in cases where, specific physiological pathways could be interesting for specific biomarkers or for ADME property prediction. And so the solution that we've developed internally is really about creating a better way for understanding the interactions between compounds, biomarkers, and physiology by using an approach that integrates machine learning and deep learning with mechanistic models. So another way to think about these two areas, mechanistic models and machine learning, is as a knowledge engine and a research engine, where the knowledge is the science that we know. And some are fluid dynamics. We know things like blood flow rates into different organs, how vasculature connections work, and how we expect things like oxygen levels or distribution to propagate along this vasculature. It can be liver enzyme interactions like CYP3A4, CYP2D6, and the effects that those have on ADME properties. And then the research engine characterizes the novel interactions, the things that we don't know as well, or might not have experimental evidence to support, the unknown unknowns, so to speak. And one example of that could be penetration of compounds across the blood-brain barrier, where we don't know exactly what mechanisms actually contribute, or specifically how these mechanisms differ between preclinical stages of development in vitro technologies like MDCK cell lines that overexpress specific transporters versus preclinical subjects like rats, mice, and cynos, and then the actual human subjects. So what we've really developed internally is a way of integrating these two engines together. And at the end of the day, we can de-risk R&D decisions by predicting the clinical benefits of these new drugs before we ever get into human clinical trials. And this approach has successfully been applied to three very diverse areas of therapeutics: small molecules, large molecules, specifically monoclonal antibodies, and re-engineered viruses. I'll go through those examples shortly. And in many of our interactions and the engagements, everyone asks, what are the inputs that we use into this modeling approach and into our platform? And the real answer is that we can take data from any stage of drug development, spanning from the very early discovery stage structural data sets, just understanding how this compound actually looks, smiles data, all the way to clinical data. And what we do is really extend our existing models to tie together any of these data sets. If we have in vivo preclinical data and in vivo clinical data, we look for integration of the research and knowledge engine together to be able to deliver an accurate prediction. As we augment that with in vitro data, it can be for the same group of compounds or for novel compounds, this platform learns and can be expanded. And now we have a more comprehensive framework. And the idea is at the end of the day, this approach democratizes these powerful tools like machine learning and mechanistic modeling. We recognize that most people that are actually doing the hands-on research aren't going to be modelers or ML experts. So we package these tools with the scalability of cloud-based systems like AWS and give them access to users in a user-friendly interface with insights from our own proprietary databases and with the robust engineering infrastructure powering this overall. So to reiterate, the way the framework works is comprehensive. And this is just a simple illustration of how we work with incomplete inputs from these features. And again, the multi-compartment model in this case is an example of the knowledge engine, and the machine learning is the research engine. And an iterative feedback process leads to accurate outputs overall. And this framework is unique and novel. What this also accomplishes is a reduced black box effect. And this is a big challenge with machine learning models where they may be able to deliver accurate predictions, but the predictions are packaged and blinded. We don't really understand what's going on under the hood. And the integration of mechanistic models really enables transparency and can actually drive research because we can identify the underlying mechanisms that aren't captured adequately with just the existing scientific knowledge. And that insight is delivered directly to our users. Now, another key aspect, in addition to the approach, is the data that is used. And I mentioned this on an earlier slide in terms of spanning the different stages of preclinical and clinical development. So what we're talking about here are network effects, where data sets that are very disparate generally can be tied together and really systematically used to build out a better core model across even therapeutic areas as diverse as small molecules, large molecules, engineered viruses, or CRISPR-Cas9 therapies, and spanning all the way from pharmacodynamic outputs like biomarkers, and including pharmacokinetic data as well. So we package that with data from data partnerships, internal data sets that we've aggregated into a rich proprietary database from public sources, and proprietary experimental information. That's all at the underpinning of what the model training has actually gone through. And with this approach, we can continuously train and test models and not only identify false positives, but false negatives as well. And validation throughout these data sets, different preclinical models, in vitro and vivo, enables us to have more confidence in our predictions and effectively deliver more accurate predictions for users of the platform that are conducting drug development experiments. Now, all of this leads to, how do we address that core problem that we laid out in the beginning? And the end result is that we can cut down the amounts of drugs that get to the more expensive stages of development. Really, the stages where failure is associated with a much higher cost because there's been a much more significant investment. So it's easier to make go, no-go decisions at an earlier stage in development. And this funnel graphic identifies and just kind of highlights how we really want to narrow that funnel the later that we go and really prioritize all of that high throughput screening at that earlier stage. And so in silico platforms such as BIOiSIM enable hypothesis-driven experiments at that earlier stage. And you can see in the corner I've outlined here some of the specific preclinical and clinical outcomes that we've conducted already or are working towards. They can be as technical as specific disease models. When we talk about chronic kidney disease, diabetes, and the blood-brain barrier, where we're studying biomarkers associated with these disease conditions, the complex pathways that can contribute, and how interaction with one target along those pathways can contribute in fluctuations in biomarkers that we expect to be important. And so on these next few, I'll get into some of the case studies where this platform has been used effectively already. And they've been blinded for confidentiality reasons, but you can tell that all of these diverse case studies are still powered by the same infrastructure, the same integration of the knowledge engine with the research engine. And so in this specific case, we were working with one of our clients, which is a large pharmaceutical company, and they had just over 650 in vivo rat data sets for plasma concentration over time. And they wanted to leverage these data sets to enable prediction of compound PK, but there are different issues. The data sets aren't actually clean, so there's missing or mislabeled data. The standard models that they were using internally were big oversimplifications of physiology. So they wouldn't capture really specific insight if that's something that was desirable either at this stage or going forward. And there wasn't really a scalable simulation model for these 650 compounds. For those that are familiar with modeling techniques and things like parameter optimization, computational expense can become a big issue when we're talking about this magnitude of results. So our approach here was an automated system of database consistency checks that identified mislabeled and outlier data sets. So the data curation process became increasingly automated. And we ran simulations across all these compounds using the same model. Because, again, to avoid the problem of overfitting and developing custom models that are so specific that they're not translatable to new outcomes, we wanted to validate this approach across all 650 compounds. And you can see the results. So AFE and AAFE are fold error metrics. And for these 6 parameters, which are standard ADMET clinical outputs that are looked for, we were able to simulate approximately 95% compounds within 3-fold error of AFE, and it's about 86% within 2-fold. And so at the end of the day, we validated the fact that our model is able to deliver these results accurately in a very scalable fashion. And now these data sets are integratable into the other machine learning prediction algorithms that we're using, and can now, for the 651st compound, 652nd compound, insights from these models can be used to inform what the next clinical stages or preclinical stages of testing would be. Moving on to large molecules. So again, this is a collaboration with with one of our clients, a large pharma company, that had over 100 data sets of monoclonal antibody pharmacokinetics, predicting a specific parameter associated with ADME. Now, the problem was that they wanted to screen for clearance earlier in development. And that's an important metric for monoclonal antibodies because it affects compound prioritizations for specific targets. So the approach is that we wanted to assess the inputs that were provided by the user and perform a structurally chemistry informed feature selection, where we identify exactly what some of the descriptors that were provided would be important and which ones wouldn't be important in the overall results, and some hypothesis-based transformations, couple that into the AI/ML infrastructure, and then again, at the end of the day, deliver the standalone user-friendly product. So now researchers there can access the power of machine learning tools without having to be explicit experts in training models manually themselves. So this is an easy-use standalone product that enables go-no-go decisions. And here's the key validation of that. So with 81% accuracy, we're able to actually predict whether compounds that are monoclonal antibodies with relatively small data sets, we're talking about 100 compounds, compared to typical drug discovery machine learning that are working in the thousands. These are small data sets. And so the ability to get this accuracy that's this high is pretty impactful. And we're really excited about these results. And we're actually trialing the software. There are a few licenses that will be in use. But again, now this serves as kind of the nucleation point where models can continue to be developed and further insight can continue to be gained using this type of model as the starting point. I mentioned reengineered viruses previously. And for context, these are drug delivery type mechanisms or even directly drugs that are becoming more prevalent for cell therapy or targeting infections by bacteria. And there are a lot of behaviors that could be present. In vivo data sets are small. So we need a way to measure actually what's important, really to understand what's important to measure at early stages in development and to characterize the relevant behaviors. There are a lot of possible hypotheses out there, a lot of publications and data sets that have been generated, but there isn't a holistic framework for integrating them and understanding exactly what's important in each context. So the approach here was to develop a model of virus-host interaction and really use that model to drive an understanding of what parameters are important and what in vivo outcomes can actually look like. Then we validated that with some existing data sets, working with one of our clients, a mid-sized biotech company. And then we're able at the end of the day to predict outcomes in vivo and predict possible interactions with even complex systems like the immune system. So the result is the ability to simulate these new outcomes and again, target specific in vitro experiments that could be used to screen these different viruses at earlier stages. And it again creates that nucleation point. So now as these companies approach development or high throughput development of new candidates like are common in AAV or bacteriophage development using either directed evolution or rational design approaches, this all feeds back into the platform and delivers a better prediction at the end of the day at an earlier stage. And now the final case study, it's highly relevant. The one thing that's clear is that for COVID-19, there's a huge need for therapies. And we actually applied our approach to repurposing existing drugs. So it's a rapid identification and simulation approach to identify which existing drug candidates could be most susceptible or most likely to help in the case of COVID-19 using existing data. So you can see on the right, there's this outlined comprehensive framework and approach that could be used in these contexts, starting from simulation of drug pharmacokinetics all the way to interactions between the viruses and the compounds at the site of action. And the key hypothesis driving this is that compounds repurposed from pulmonary hypertension that target ACE inhibitors and calcium channels, specifically calcium channel blockers, could be utilized to prevent some of the cytokine storm effects, in addition to mitigate some of the viral infection. So the results here are pretty straightforward. We were able to simulate the pharmacokinetics of drugs accurately, and we've submitted the manuscript. It's currently under review, but we were able to simulate the experimental data well and also predict penetration to these sites of actions that are implicated in COVID-19. And so we expect that the next logical extension will be, once we have confirmation of the mechanism of action, we can use this approach to quickly conduct first-in-human simulations because of what we've already verified the translatability aspect of this drug disposition. So what we've done, and really the end way and how we interact with our different customers and users is actually pretty flexible. So we customize the model and the infrastructure to meet the data needs, the AI needs, the modeling needs, and the commercial deployment needs of our different users, where data can be hosted either on our infrastructure and some hybrid cloud methods. The AI/ML models can be trained internally by us or in collaboration with our different users, and we develop these custom models. And again, every model that we develop is validated across the comprehensive framework and datasets that we've designed internally as well. And so our CEO, Dr. Joe Varshney, is actually on the next panel. So if you don't get a chance to ask me all of the questions that you have today, feel free to reach out to her. But at the end, the goal is that we enable users and researchers to leverage computational resources in your own companies and empower you to work with these systems. So thank you very much, and I'm happy to take any questions at this time.
Victor, very nice presentation. Thank you. We have a question from Harumi Tada. His question is, for COVID, weren't ACE inhibitors already identified as potential repurposed drugs without the use of AI? And aren't there data to suggest that ACE inhibitors do not influence the outcome of COVID?
That's an excellent question. So for COVID, we were looking at actually not just using ACE inhibitors, but also ACE inhibitors coupled with calcium channel blockers. And there are different specific subject populations that these therapies may work in, or actually, in some cases, they may not help with the disease condition. The idea is that we can use the combination of both, so calcium channel blockers, so calcium channels can be hijacked by viruses for entry into the cells. So the hypothesis is that these combinations can be used together for mitigating some aspects. And to be completely honest, we may find that these combinations don't work in some healthy patients, but they may be more likely to work in patients that have pulmonary hypertension or some other conditions where inflammatory side effects in the lungs or the nasal cavities or the guts can be more significant. So at this point, there definitely needs to be further confirmation of the mechanism of action. There's definitely, in addition to ACE inhibitors, there are ACE2 inhibitors that are being developed. And again, the key point is that this is that nucleation point. Once that mechanism of action if that mechanism of action is validated, now we have a rapid repurposing framework. And we can translate this to other targets as they're identified to be implicated in the disease.
So I would agree with you that combinations make a lot of sense. And I wonder, are you able to model more than two products together? And if you take a look at what happened with HIV and AIDS, ultimately it was the cocktail that became successful. And I think there was at least 3 and sometimes maybe 4 or 5 different drugs in a single pill.
Absolutely. And the idea is that we can model really as many species that we expect to be implicated. So this is where the pharmacodynamic side of the modeling integrates with the pharmacokinetics. So when we have, let's say, 5 drugs or even 10 drugs, we simulate their distribution to sites of action, understand if there are any potential drug-drug interactions between them, and then look at the specific targets that they're interacting with and map those together and see if they really impact the disease condition that we expect it to impact. So whether it's infectivity and replication of the virus, or if it's the inflammatory side effects. And now we have a network-based model. It combines different data sets. It can be looking at those 5 or 10 drugs in isolation, and then combining them together to understand what that network effect is, or any combinations thereof.
So one other follow-on question, if I may, is are you able to look at underlying diseases that patients may have, like, perhaps a drug works differently in a patient with diabetes versus a patient with heart failure versus, person with asthma? Are you able to look at things like that?
Absolutely. And we've actually developed internally a diabetes model that looks specifically at how interactions for patients with the condition versus healthy patients can happen with intake of glucose or intravenous glucose tolerance tests. So definitely the same approach can be applied to patient stratification, where we can translate, as I mentioned before, between different species, and we can also identify how within a specific species, different genders, races, or diseased populations can also vary in their effects and responses to different drugs. So whether it's, again, pharmacokinetic, or there's some biomarkers are associated, for instance, inflammatory biomarkers, or it can be, some diagnostic biomarkers as well.
Okay. Victor, that's all the questions that we have from the audience. I want to thank you very much for your presentation. It was well done and very insightful. Appreciate your participation in the summit.